There is a wasp in the tropics the size of your thumbnail. It is iridescent green, almost beautiful, and it is capable of performing precision neurosurgery on a living creature, not to kill it, but to keep it alive. Docile, compliant, a walking nursery for its children. No anesthesia, no scalpel, just a stinger shaped like a hypodermic needle, guided by what researchers are now calling the most sophisticated targeting system in the insect world. Its name is Ampulex compressa. You likely know it as the emerald jewel wasp, and what it does to a cockroach, step by step, injection by injection, is one of the most precisely calibrated acts of biological manipulation ever documented in nature. Wait, that's real. Let's walk through what actually happens, because the sequence matters. The jewel wasp targets the American cockroach, Periplaneta americana. It's 3 to 4 times the wasp's own body weight. In a straight fight, the cockroach wins, so the wasp doesn't fight.
Stinging it twice. The first sting disables the roach's front legs. The second, she delivers directly.
It operates. Step 1. The wasp delivers a rapid sting to the cockroach's midsection. This isn't the main event, it's the setup. The venom temporarily paralyzes the cockroach's front legs for approximately 2 to 3 minutes, just long enough to immobilize the target. Step 2. The wasp positions itself head-to-head with the cockroach and inserts its stinger directly into the cockroach's head. What happens next took researchers decades to understand. The stinger doesn't inject venom randomly, it probes. Studies using high-resolution imaging at Vanderbilt University showed that the wasp's stinger contains mechanoreceptors, sensory organs that detect differences in tissue resistance. The wasp is, in real time, feeling its way through the cockroach's brain, searching for 2 specific neural clusters. The first target is the subesophageal ganglion, a neural structure that controls the escape reflex. A precise injection of GABA-like compounds here suppresses the cockroach's ability to initiate voluntary movement. Not unconsciousness, not paralysis. The cockroach can still move. It simply has no drive to escape. The second injection targets the supraesophageal ganglion, the primary brain structure associated with complex motor behavior. This injection reduces dopamine signaling, suppressing spontaneous locomotion. The total operation takes under a minute. The result? A cockroach that is alive, responsive to external stimulation, capable of walking, but will not run, will not fight, will not flee. It will simply stand there, waiting for whatever comes next. What comes next is this. The wasp grabs the cockroach by the antenna, like a leash, and walks it back to a pre-prepared burrow. The cockroach follows, willingly, or rather without resistance. It has the physical capability to bolt. It doesn't. Inside the burrow, the wasp lays a single egg on the cockroach's abdomen. Then it seals the entrance. The cockroach will remain in this state, alive but docile, for 3 to 5 days while the wasp larva develops. When the larva hatches, it chews through the cockroach's exoskeleton, enters its body cavity, and systematically consumes the internal organs, starting with the least critical, extending the cockroach's life, and therefore the freshness of the food supply, for as long as possible. This is not random. The feeding sequence appears to be ordered. The larva consumes the digestive organs last. Researchers believe this may be an evolved behavior to prevent the release of enzymes that would accelerate decomposition. The cockroach is alive until near the very end. Now, here's where the neuroscience gets genuinely startling. In 2018, a team at Ben Gurion University of the Negev published findings in Current Biology showing that the wasp's targeting process isn't instinctive in the way we'd assumed. Naive wasps, those with no prior hunting experience, still successfully locate the correct injection sites. But wasps whose antennae had been surgically removed could not. They injected widely and produced zombified cockroaches less than 35% of the time. Then in 2025, a Rockefeller University team produced the most detailed connectome map of the cockroach subesophageal ganglion ever completed. Their findings were published in Nature Neuroscience. The map confirmed that the wasp's venom targets what they called escape command neurons, a specific cluster of approximately 60 cells whose activation drives the flight response. 60 cells. The wasp finds them in under a minute, in a moving target, with a stinger. For context, human neurosurgeons preparing to target a similar neural structure in a patient would rely on fMRI mapping, stereotactic frames, and intraoperative monitoring equipment worth hundreds of thousands of dollars. The wasp does it by feel. So why does this matter beyond being a spectacular piece of natural horror? Because it's changing how neurologists think about targeted drug delivery to the brain. The venom cocktail used by the Jewel Wasp is chemically quite simple. It's primarily gamma-aminobutyric acid, GABA, and a taurine derivative. These are not exotic compounds. What makes them effective is the delivery. Precision placement at a specific neural cluster at a specific concentration in a specific sequence. The lesson neurologists are extracting from this is not copy the wasp. Its location and timing within the brain are potentially as important as the compound itself. A drug that floods the entire brain may produce systemic side effects. The same drug, delivered precisely to a target ganglion cluster, may produce a localized effect with dramatically lower collateral impact. Research groups at Johns Hopkins and ETH Zurich have begun studying the wasp's mechanoreceptor-guided stinger as a model for next-generation neural probes, instruments that could navigate brain tissue by resistance feedback, the way the wasp's stinger does, rather than relying on pre-surgical imaging alone. The wasp has been doing this for roughly 25 million years, based on amber fossil records. It has had longer to optimize the procedure than we've been practicing medicine. Here's the thing about the natural world that I keep coming back to. Every time we look closely enough, really closely with the right instruments, we find that biological systems have already solved problems we're still struggling with in our labs. Not because nature is smarter than us, but because evolution is the longest-running R&D program in history. 25 million years of iteration is 25 million years of failed experiments that didn't survive, and successful ones that did. The Jewel Wasp didn't set out to teach us anything about neurological drug delivery. It set out to feed its larvae. The fact that its solution maps onto one of the most difficult challenges in modern medicine is a coincidence of convergence. Two very different optimization processes arriving at the same insight from different directions. That's what makes biology worth paying attention to. Not the gore, though I understand that's part of the hook. It's the fact that the answers to problems you didn't know you had are hiding inside systems that evolved before we were here to observe them. The wasp has been performing successful brain surgery since before the pyramids were built. We're just starting to read the paper. Subscribe to Wait. That's real? New deep dives every Wednesday.